So today I kind of finally got an overview of um, the company as a whole. Because of the non-disclosure agreement, I guess I'm not allowed to say any specifics, but they make two, or they're researching slash developing two different cancer drugs, and then another one, I think it's for like arthritis or something, and they're all monoclonal antibodies, which are proteins. And okay, so there's always like the first version of a drug, and that gets a patent for a certain number of years, and the company that makes that can charge, however much they want, and that's why a lot of these really new drugs are so expensive. So companies like this one also do work, well, they call them biosimilars, but they're just like generics of these monoclonal antibody drugs. So yeah, there are different groups. Like, I was in molecular biology this week, and so the day before I think they're called, you impregnate a petri dish with some of the E. coli. Okay, so they created their plasmid vectors with this special gene inserted in it, and then those get transmutated, I think, into these special E. coli cells, and these E. coli cells are called competent cells, which means that they are able to take outside DNA and put it on the inside and incorporate it into their own genome. I think I've said this before. So then we impregnated a petri dish with a special type of antibiotic in it, so it killed. And when this plasmid also has another gene in it that codes for the resistance gene for this antibiotic in the agar plate. The ones that have successfully incorporated that plasmid into their genome are the ones that survive on this agar plate. And so we incubated it overnight, and then Thursday morning we picked them out. So the person that I'm working with, she picked out 10, and then I picked out 10, and then they have to. It's quite difficult. The colonies are extremely small, but yeah. Mine turned out better than hers. Mine, after 1 hour of incubation, grew a lot, and hers were barely there at all. But I think they pretty much evened out in the end. And then I don't think it's called freezing, but we stabilized them by putting a special solution, and now they're being stored at 4 degrees Celsius until, so they can just stay where they are right now until they use them next. I think some of them are going to go to be sequenced to make sure that their genomes are completely correct. And then yesterday afternoon, I don't remember what I did. I think we finished off our inverse PCR, and they started a process called the Western blot, and it's basically a way to separate a bunch of proteins that are mixed together. In this case, they're separating their drugs with other proteins that might be in that serum. And first you use gel electrophoresis to separate out the proteins, same thing as the DNA version. And then you transfer the bands onto this special membrane. And then today we took the membrane and you make it glow, and then you expose it to a plate and basically transfer the image onto this special plate, and it's in a dark room, of course. And mine turned out very well as well. They used mine as a secondary one. Each of I think there were 6 of us in there, 5 of the workers, and then me, and mine was the second best, which was nice. And then today the molecular biology person was not here, so I got to go into the cell room, and I'm going to start another one because I think that would work.